My name is Lauren Horton. I worked on this project along with Shelley Kozlowski and Carol Franklin. You'll see them later on in the video, but I'm going to go ahead and get us started. Our differentiation project focused on a high school biology class with an emphasis on the gifted population of students. The three areas of differentiation we focused on, as you can see on our infographic, are content, process, and product. I'm going to dig into content with you in a little bit, whereas Carol and Shelley will focus on the process and the product. The content is really focusing on what is being taught. Of course, we always want to meet our standards, but with our gifted population, we kind of want to make sure we go beyond the standard and offer extensions where we can get them to that distinguished level of learning. You can see we have 3 different research articles with easy to scan QR codes to take you to the direct article, as well as a brief summary so you know what you're getting into. We have one article focusing on the cost effectiveness of differentiation, as well as getting teacher buy-in into using differentiated strategies. This focuses on the biology classroom, specifically in ecology and genetics units. We also have an article on differentiation for gifted students. This article includes lots of great best practices and examples that teachers can use easily in their classrooms. And finally, one of my favorites, an analysis of differentiation in the high school science class. They use both qualitative and quantitative data to show the effect size of differentiated instruction. And it might be a spoiler alert, but differentiation definitely increases student learning. On down our infographic, you can see we have our standard that we're focusing on, which involves one of my favorites, gene expression, with a focus on DNA and RNA. Here, you'll see later on, and Shelley and Carol will go into the process and the product later on, like I mentioned. But for content, you can see we're really taking our students from the proficient level to the distinguished level. If you click here, you'll go to the Georgia Standards of Biology, whereas here, this will take you to our differentiation infographic on learning targets. For content, it's really important to focus on going from one target to the next. Everyone starts out as a developing learner, and this is where your vertical team will come in handy. You can look at what the students are supposed to come in with their previous knowledge. In the area of DNA, you can see they can just identify the types of mutations. Proficient is where the standard wants them to be in a high school biology class, not just identifying mutations, but also looking at the negative, positive, and neutral impacts. Whereas going from this to the distinguished level, we're really going to focus on taking it beyond that standard and linking other subjects or synthesizing information. In this example, I add the word make predictions and looking at how the entire gene pool of the population can be affected based on the mutation rate, which really gets us into the unit of evolution that they'll come back to. So I hope you enjoy the rest of this project with Shelley and Carol. This was a great time learning lots of information. Thank you.
Hi, my name is Carol Franklin, and I will be sharing with you how to differentiate process for gifted learners. Differentiating process refers to changing how content is taught. It also refers to how students make sense of or understand the content. Teachers may vary instructional activities to suit learning styles, language proficiencies, or cognitive levels. Examples include using graphic organizers, hands-on experiments, small group discussions, or interactive activities that allow students to explore content in ways that align with their particular strengths and needs. 2 best practices for differentiating process for gifted learners are to utilize small group, which requires students to explain their thinking to peers, evaluate their problem-solving approaches, and adapt based on feedback, and problem-based inquiry learning activities, which focus on open-ended problems without a single correct answer, pushing learners to develop critical thinking and to be innovative and create innovative solutions. An example of differentiating process would be a lesson on structures of DNA and RNA. The standard is SB2, obtain, obtain, evaluate, and communicate information to analyze how genetic information is expressed in cells. Part A of the standard says to construct an explanation of how the structures of DNA and RNA lead to the expression of information within the cell via the processes of replication, transcription, and translation. Engage. The activity would require that students be divided into small groups and presented with a real-world genetic mystery or an ethical dilemma related to gene expression. And this should take about 10 minutes, or it could be a little longer if it needs to be. A fictional population suffers from a mutation in the hemoglobin gene. How could this mutation arise, and what strategies might scientists use to correct it? The discussion questions that students would be asking in their answering in their small groups would be, how do changes in DNA or RNA affect protein synthesis? And why is understanding replication, transcription, and translation crucial for solving genetic problems? The resources would be a short video clip from DNA to protein, and a case study handout outlining the genetic mutation scenario. For the explore phase of the lesson, it would also be inquiry-based investigation, and it should take about 25 minutes or maybe a little longer if needed. The hands-on task that students will be given, their small groups will explore the molecular process by using interactive simulations, learn genetics, DNA to protein simulation or DNA workshop, or model kits, molecular modeling kits for DNA and RNA. Challenge questions would be, how do structural differences between DNA and RNA affect their replication, transcription, and translation functions? Also, how might an error in DNA replication propagate through transcription and translation to affect protein function? The resources or materials needed would be Chromebooks or tablets for accessing simulations, molecular modeling kits, diagram handouts of DNA and RNA structure.
Hello, and welcome to my portion of this differentiation project. I'm going to be focusing on the product differentiation. My name's Shelley Kozlowski. So with product differentiation, this is changing up how students are assessed and how they learn the material. And so with product differentiation, looking at this portion of our graphic, it's changing up how it is assessed. And so this relates to the ways that students demonstrate what they have learned. Teachers can offer choices in the format of assignments or assessments to showcase understanding. So for example, instead of a traditional test, ELL students might create a poster, present orally, or complete a project that reflects their grasp of the topic in a format that they find more accessible. And so with our gifted population, this is a way, especially for the creative gifted student, to show their knowledge of the standard. And again, we are assessing standard SB2A, which is to construct and explain how the structures of DNA and RNA lead to the expression of information within the cell via the processes of replication, transcription, and translation. This is my portion of the infographic down here, and it's talking about the product. So getting into it, this link takes us to the standard again, and then this is just one example of how a teacher can differentiate product. I chose the choice board and also a tiered project with real world application. So with a choice board, you can see here that there are several different options that a student can take. They need to make a tic-tac-toe in some way in this choice board, so they can choose this direction. A lot are going to choose the free space option. But you can see here, this ranges in making comic strips to coming up with a project proposal, a TED Talk presentation, writing poems, developing games, designing a lab, all good options and different ways for students to show what they know, and in different ways for product. The tiered project starts with a case study development, starting with some research, and it will include visuals. Then they move on to the proposal. This is where they write a grant proposal for a biotechnology company addressing a genetic challenge. And then they take it to the public with a public awareness campaign where they use multimedia and social media like Instagram, things that they already know, to gain public awareness. And then there are ways to not just differentiate product, but going to the graphic, the materials and resources that the teacher will need. And these include things like, zooming in here, things like access to Canva or Google Slides for the visuals, lab materials, also research resources and some video creating tools. For the tiered project, they're going to need access to some guidelines and some ways to organize their research, maybe a template. For the grant proposal, they're going to need some STEM related grant proposal templates, tools for multimedia campaign research like Adobe Spark or PictoChart, and then finally some presentation tools like Google Slides, PowerPoint, Prezi, and not mentioned here, but our favorite, Canva. And then finally, teachers are going to need to have rubrics, some very clear guidelines for how the gifted student is going to be graded. I find that my gifted students are very receptive to the rubric and how they can make the best score possible. And I broke this down into AI tools versus non-AI tools. The AI tools include things like ChatGPT and Magic School AI, Rubistar, and then the non-AI tools, your Google Sheets, your Google Docs, which already have templates built in for rubrics, Fluency Tutor, and Kidem. So all these are ideas for product differentiation. Thank you for your time.